Good afternoon and welcome. We got a lively group here, so let's all settle down for the main event. Thank you, thanks. My name is Jamie Williamson. I'm a professor here at Scripps Research, and I'll be your host and moderator today. I want to start, we have my slide. Thank you. So first I want to welcome all of you. We have looks like about 200 people here in person. And I'm told we have 4,000 attendees in the virtual audience, so welcome to all of you. I know many of you tune in from around the world to hear these lectures, so it's great to have everyone here. I'd also like to acknowledge our Science Trust and Legacy Circle members, and we thank you especially for your support. So today, the primary purpose of today's lecture is to inform you about the science of vaccines. How do you discover a vaccine? How do you invent a vaccine? And where do they come from? And I think we want to give you a look under the hood of how this happened. So you're going to hear a remarkable story today of the genesis of the COVID-19 vaccines. But I think what's even more exciting is that you're going to hear what's going to come down the road. So I think the threat from infectious disease is never going to go away. New pathogens are always going to be emerging, and the process of vaccine development has been very slow and very time-consuming, and yet we've learned so much, especially in recent years, about how to accelerate this. And our speaker today, Andrew, is at the forefront of accelerating these discoveries. So how can we prepare for what is inevitable to come, which is the next pandemic? Now, I want you to listen to what Andrew's having to say, but I like to recommend active listening, where you're constantly asking yourself, what would be a good question to ask? We're going to do Q&A at the end, but just listen actively and try to follow along and invent questions that we can come back to at the end of the discussion. Now, Andrew was a student here, so I've actually known him for almost, I think, 20 years at least. He became a faculty, and then he became sort of an institutional hero. So you're going to hear about his personal journey, how the environment at Scripps led to this remarkable program of vaccine discovery, and I really think you're going to find it astonishing what's coming down the pipe. So there's an elephant in the room. Executive orders have placed some real fearsome constraints on science funding in the country. And it's bound to really adversely impact the whole science infrastructure. And Scripps is not alone in this. UCSD, Salk, Sanford Brennan Priebus, we're all in this together in California, in the United States. Everybody is going to be impacted on this. And so we need your help. And there are a variety of ways that you can help. You can be engaged with our philanthropy group, but you can also become an active advocate for science in the country. It's really critical, the things that go on here and at other institutions. It's groundbreaking discoveries, generating new medicines, clinical trials. There are people who are having their treatments stopped mid-course. It's the discoveries for the next medicines, and it's also the science infrastructure. We're training the scientists who are going to go off into the pharmaceutical industry and biotechnology industry and create new medicines. We have to get through this, and we sure could use your help. So let me introduce Andrew. So he's a professor in the Department of Integrative Structural and Computational Biology. He's my colleague. We know each other quite well. We occasionally have board meetings, surfing together, which is great fun. And I'm going to come back at the end of Andrew's lecture, and we're going to have Q&A. So for those of you in the audience, There'll be lightsabers so that I can call on you. So raise your hand and someone will find you. If you're in the virtual audience, you can type your questions in at any time during the lecture into the Q&A or the chat. And I have an iPad and it'll get relayed to me at the end. So we're looking forward to discussion, active listening, lots of questions, exciting science, and most importantly, Andrew.
Thanks, sir. All right, we ready to go? I think so. All right, here we go. So first and foremost, thanks, Jamie, for the kind introduction, and thanks to you all. It's actually quite flattered that so many people showed up, and it's a different audience than I get to talk to. And so, my story and journey is really all about, as I say here in the subtitle, mentorship, state-of-the-art technology, the trainees I've had, collaborators, and really the Scripps ecosystem shaping my career, who I am today, and really enabling in putting me in a position to contribute to vaccine research and COVID vaccines. And so I'm going to tell that story mostly through the lens of the people that contributed and the advocates and all of the infrastructure that it took to get to this point, because I didn't start out trying to make a COVID vaccine or contribute to one, in fact. So first, I'd like to just acknowledge that's the most recent picture of about 3/4 of my lab right out here in the courtyard. And it's a fantastic group of diverse scientists, all contributing in different ways to the program. And I could stand up here and tell a story about each one of them and their amazing contributions and how amazing they are as people. Today, I only have a limited time, so we're only going to hear about a couple. I just want to acknowledge that in addition to trainees, which come in for a short period of time, a graduate student, a postdoc, I've been really fortunate to have people that have spent almost their entire professional career with me, coming right out of an undergraduate degree and now 10 years or more in the lab, and really the core engine that runs the lab and allows us to do all the things that we do. Behind them, there's a massive infrastructure, right? Science doesn't just happen with those people. There's administrators, there's people in the finance and grants department, there's philanthropy and comms and legal, business development. All the elements that are encapsulated by science takes a village, and a village, much bigger than this. And you'll meet some of them when you interact here at Scripps. So we've been very fortunate over the years to also get a lot of support from the NIH, the National Institute of Allergy and Infectious Disease. We've been part of large consortia and centers, and we're increasingly recruited to participate in a lot of these vaccine efforts that take everything from the basic research scientists to the clinicians and beyond, and the clinical trial organizers. And so that's really what helps build such a big lab, buy expensive toys, and do the science that we do. So to wind it back, I'll start, I grew up in suburban Boston. I got my first chemistry kit when I was, I think, 9 or 10 years old. And my mother famously said, "You'll either be an arsonist or a scientist." So I think I chose okay. But then importantly, I made really a rather uninformed decision at the time, mostly because I like Duke basketball, I think, to go to Duke University. And it was the furthest my parents would let me go and the best school I could find. I wanted to go to Stanford, but that was out of the question. I fortunately ended up in a lab as part of a work-study program to help pay for college with Michael and Mary Reedy. And they really just took me under their wing and inspired me to be curious in science. We worked on these giant water bugs from Thailand, and the image you see in the back is the flight mussel. And the flight mussel image with an electron microscope reveals all these molecular details that help you appreciate how mussels work, and it was really basic. But it was a phenomenal experience. And I took so much from that. And then at the end of school, they said, well, what do you want to do next? And I said, I don't know. I don't want to go to med school. I like research, but I don't know, what to do next. And, I was in Mary's office, and she picked up the phone, and she called Ron Milligan. And she said, this guy, Ron Milligan at Scripps, is doing some really cool stuff with something called Cryo-EM, and it's going to be the future. So this was around 2001. And I said, what do I have to lose, right? So with that phone call, Ron offered me a job, and I moved out, and really sort of the rest is history. That's Ron and I at a Gordon conference discussing science over a beer, as we often did. In his lab, I learned a lot about Cryo-EM and the technology. We took microscopes apart, we put them back together again. It was pretty casual. And my science revolved around antibiotic or multi-drug resistance in bacteria and cancer. And so it was really a structure, function, mechanism lab. And then that progressed into a short postdoc here with Jeffrey Chang. And then a faculty position sort of magically appeared for many reasons. And I kind of filled this niche in this membrane protein world, and I was sort of set off on that world. In addition to Ron and the Reedies making these formative impact on me, I really have benefited from the Scripps faculty at large. So Ian, Dennis in the vaccine and antibody space have really inspired and pushed me, and Ian has been a huge advocate of building up the infrastructure that now exists here. And Jamie has just been sort of a guiding light throughout all this. He famously told me to not accept the offer at Scripps and to take a position at Stanford. I said no. And then when I almost left Scripps, Jamie's the reason I stayed here. It's wonderful to have these people, and this is just a small subset, but these people really, are your sounding boards. They influence the scientific directions you take, and have really, supported me throughout this journey. So let's start. So Ron used to like to quote Richard Feynman, who is brilliant, and I would totally recommend going on YouTube and just spending some time, because Richard's one of the finest scientific minds and thought beyond his years for sure. And in 1959, he said, it's easy to answer many of the fundamental biological questions. You just look at the thing. Make the microscope 100 times more powerful, and much of biology will be made easier. And he said, and of course, biologists will be happy because then they won't have to do math. As I said, I landed at Scripps in 2001, right about the time that Ron, Clint, and Bridget were putting together this cutting-edge molecular microscopy center called SymBio, which was down the road, is no more. And it really was understanding the potential of the electron microscope and putting in the thought, work, and effort to build up the infrastructure and be poised to take on difficult biological problems by putting it in a microscope. But as Ron notes, it's not routine, it's tedious. There's only probably 20 labs in the world working in this space. We were called blobologists because we weren't getting really nice pictures. But they created this amazing fertile environment where they were thinking ahead into the future. And we used to have semi-annual meetings with basically the whole field would come to Scripps, talk about the future, and we would have routine run-ins with Jacques, Joachim, Richard, eventual Nobel laureates for electron microscopy. And we sort of didn't even appreciate how great it was at the time. In retrospect, you can appreciate how fertile of an environment this was. And the careers of most of my colleagues and trainees have been really amazing and impactful all over the world because they were part of that environment. And that's really where the environment is shaping you, because people with foresight and thoughts have invested in that. And Scripps was convinced to buy these what were at the time relatively low resolution but expensive toys for Ron and Clint and Bridget to play on. So 1930s, the electron microscope was invented by Ernst Ruska, It hasn't changed much in terms of the optics. It's got a lot more, plastic, and it looks a lot nicer now. A lot of the groundwork was done, in the 90s, 2000s. And then there's the advent of something known as a direct detector. So normally, when I was doing my graduate work, we were collecting on film. So we were converting electrons from the electron microscope that would go through our sample, be scattered. We would convert those into photons, and then we would take film images. The problem with that is, even though you've embedded these samples under liquid nitrogen temperatures and they're really, really cold and not moving around, in fact, they are moving around in the microscope. So if you look at the bottom image, you can really appreciate what a rotavirus looks like in a conventional either film or one of the early digital cameras. On the right, you can see how crisp and clear it is by being able to detect electrons and also collect movies. So you're collecting these movies that actually track the motion, and that's the top bit there. You can see the vectors of these particles moving in ice. And it's really analogous to what an iPhone can do right now, right? So this picture, one of the best pictures other than my kids I've taken. I was literally walking out of the grad office here. I saw a hawk. I pulled the camera out of my pocket and snapped that picture of a hawk mid-flight, which you can see the details of the tips of the wings and so many details. Even though I was moving, it was moving. And then also it was perfect because John, that's John Torres, he's a lab manager. He's been with me his entire career. He is the lab photographer, and you'll see some of his pictures today. But I sort of switched the script on him totally by accident. But essentially, that's why we get pictures of kids moving and hawks moving, and we can do that now at an atomic level with these special cameras. And Scripps made the investment very early on for one of those first detectors. So since then, we've been busy and expanding. So this is the current Cryo-EM suite, and I know there'll be some tours offered if people want to walk just across the courtyard and see. First and foremost, to acknowledge, it's not just hardware. There are people, Bill, who recently retired, Hannah and Will, who help run this facility. It's not a trivial amount of coordination to run this facility and stay on top of the maintenance and all of the moving parts behind the scenes. But the TechNI Spirit is now the only microscope that existed when I was a student. We've now replaced and upgraded with a number of these amazing machines. And it's one of the biggest and best in the country for sure, if not the world. And it's really about over the course of the years, over a $30 million investment just to keep this going, and several $1,000,000 a year just to keep it up and running. To give you a sense of scale, that's our Titan Krios in the middle. Once we solved the first structure of HIV envelope, the National Institute of Health and Scripps split the cost for this state-of-the-art microscope in 2014, which is 10 feet tall and does amazing things, including, that's my daughter when she was 2. We put her in the microscope. Pretty good resolution. All considered a wary grandmother hand, just so we didn't void the warranty on $7 million instrument. So that's what we got now. And then on the control room, here's a credit to John. He makes me look good, so I never collect data or sit in front of the microscopes like this. But John Cohen, who is a local science writer and an amazing advocate for vaccine research, HIV, and a lot of things, and fellow surfer and friend, wrote this article called The Dream Vaccine. And instead of putting a science picture in, they put me in there, which was really nice, and John made it look really nice. Behind the scenes, the things you don't see are a huge investment, multiple millions of dollars in high-performance computing. As you can imagine, we do a lot of calculations and math behind the scenes. JC, who's our HPC manager, and then Charlie, who's the architect in my lab that built this this engine to collect data, process data, and store data, which is now a huge asset in the age of AI and machine learning. And having this behind the scenes is really critical to the things we do. And to sort of give you a comparison, a real world comparison, again, when I was a student, I would be lucky to collect maybe 200 pieces of film in a day. We'd have to print them out. Now these cameras can collect 1,500 frames per second. So that's terabytes of data every day. We, of course, don't print them out, but if you did, you could reach the height of Mount Everest in a few hours, and Charlie compresses that into 7 inches, which is pretty remarkable. Not only the compression, but also the access and being able to do something useful with the data. So why do we do it? What's the sort of end product? It's this, right? This is in gray. Is the actual data that comes out, the structural information, these maps that then experts in structural biology interpret. They place amino acid side chains. They know where to put carbon, nitrogen, oxygen, other molecules, which then reveal at the atomistic level how an HIV antibody neutralizes HIV, for example. And we can use that for all sorts of engineering purposes, and I'll kind of hit on some of these. But that's just, really the beauty of what we are observing at the micro scale. So vaccines. Why do we work on vaccines? So vaccines have had a disproportionate effect on society if you look at, whether it's just, in terms of mortality, but also morbidity. And you can see this is a nice chart put together by the World Economic Forum that, about 1 billion lives have been saved with the advent of vaccines. And really, that in clean water and toilets and a few other things are really the key things that have allowed us to actually survive a lot of plagues. So, the estimates for the COVID vaccine vary widely. And as I note, modeling, mathematical modeling really requires robust access, robust public databases, which is really critical, and we take for granted often to do this. But anyway, so between 2 and 20 million lives saved in the first year from COVID vaccines. Another study, it says it's about 4 times as many deaths and over $1 trillion saved just by the implementation of the vaccine. And then on a sort of related note, viruses do a lot of things, not just make you sick and give you a cold or a fever. For the HPV, for example, human papillomavirus, it can cause cancer. And so HPV vaccine is one of the most remarkable success stories because of what it does. Not only does it prevent you from getting HPV, it has resulted in a greater than 90% reduction in cervical cancer and other related cancers. So things that happen now, a viral infection, can manifest many, many years later, and this vaccine protects against that. And the additional data that's really nice about this is they've gone into communities and looked at the vaccinated versus unvaccinated incidence of cancer, and the unvaccinated incidences are also dropping, and that's because of herd immunity. Enough people are protected that HPV isn't circulating. I think there's many other vaccines that probably have other effects like this, but HPV is really well studied. Okay, so what do we work on? Here's the sort of rogues gallery of some of the ones that we worked on, the things that we put in the microscope and want to understand at a molecular level: HIV, Ebola, malaria, Lassa, hepatitis C, influenza, coronavirus. And you can see each one has its own shape. We color it, make it look pretty, of course. And most of these structures are understanding how antibodies in the immune system interact with the key weak spots on viruses. And that's because we're trying to program immunity through protein design. So there's a picture of an HIV virion, and you can see on the surface it's decorated with these red envelopes or spike proteins. People many vaccines that have been developed are just live attenuated or inactivated viruses. And that's enough for the immune system to say, mount an immune response and have lifetime immunity for measles or otherwise. For HIV, it has all the immune evasion mechanisms you can imagine, and you can't just do it, right? So one of the things that Jonas Salk tried to do at the end of his career was make an inactivated version of HIV vaccine. That didn't work. So now we're trying to program the immunity more deliberately by solving structures of these key sites of vulnerability, engineering very small components, and it's the scale is really remarkable here, right? We're trying to take atomic level details of tens of thousands of atoms, engineer them to make a vaccine that can save hundreds of millions, billions of lives. And we're doing this in real time. It's pretty amazing that the technology has progressed so that we went from empiricism, where you kill a virus, you stick it in an animal or a person, does it work, to actually having very strong molecular control over the immune response. Okay, so again, we're going to go back in history. How did it all start? Like I said, I was working on membrane proteins and had no clue about vaccines or antibodies. Ian Wilson, a colleague, came to me and said, I'm doing crystallography, another structural technique. It's sort of hit or miss. You get a crystal or not. And he said, you know how to use the microscope. Put it in the microscope. Tell me why this won't crystallize. Fortunately, I had this guy Reza in my lab, postdoc, right before he left for a faculty position, stuck in the microscope. And it was pretty clear as day, right? So you can see on the screen, there's at the top there in the one, I think you can see this is the thing he was trying to crystallize, and then here's two antibodies stuck to it. But if you look at the progression of images, that thing is flexible and moving around, right? Something that's flexible is never going to crystallize. So again, we just looked at it. We said, okay, here's your problem. We get a little bit of information. We're still blobologists. We haven't given you much, but it's probably not going to work. And then that sort of got parlayed. I had two graduate students, rotation students at the time, JH and Daniel, and they became intrigued by the microscope and what we could do. So Ian was also trying to crystallize, this is just a piece of HIV envelope. He was also trying to crystallize the whole thing. This is what most of the things he was trying to crystallize look like, pretty much junk. Daniel and JH diligently worked through lots of different biochemistry and lots of different sequences of these antibodies, and then it was clear as day. All of a sudden we had this very well-behaved, triple-lobed, well-resolved HIV glycoprotein, and that was the point where my lab basically went from membrane proteins to vaccines. And so really this is sort of a bench to bedside story. So in 2011-12, we discovered, the first well-behaved envelope glycoproteins. That was the time Scripps made an investment in a direct detector. And actually on one of the older microscopes, we solved the first high-resolution Cryo-EM structure. And Ian was also now able to crystallize this in parallel. And so we had the structure. Once you have the structure, you can design, you can engineer, you can do all the things you want to do. So by 2019, this molecule was going into the clinic, or being clinically produced, and we're now solving the structure of the clinical material, which is a pretty big advance. And now we're working on really analyzing a lot of the clinical trials, because for HIV, for many of the bugs we work on, the problem isn't solved with just the antigen. We have to learn and iteratively learn and cycle through that loop. And this is, again, where it takes a village. There are funders, clinicians, volunteers that sign up for trials and allow us to do molecular medicine in real time on vaccines. And that is so critical to what we do. And a lot of our work now is developing new tools to analyze immune responses from the clinic so we can go back to the drawing board and redesign. I thought it'd be nice to include a few of these research arcs as well. So Zach came to my lab. He was a physicist from UCSD, like a physics and math nerd. And he's like, but I want to do something with viruses now. So I said, okay, cool. I put him on this project. And he solved this beautiful structure of the sugar coating of HIV envelope. So this is one of the immune evasion mechanisms. It's covered in sugars. Antibodies can't bind it really well. And he figured out a way through a lot of math, actually, how to image it at different resolutions and understand the dynamics, right? Because a protein might look static when I show you a picture, but it's moving and wiggling all around. And so he worked there. And then, so he took that experience and he jumped over and got a faculty position at University of Missouri. And he just published this amazing paper in Nature from 2025. And that's the human LDL particle. So that's the cholesterol molecule. So just like we knew HIV, we've been searching for that structure for 30 years. People have known about LDL for a long time, but they didn't know what it looked like or what apolipoprotein and the other components are. And so again, Zach used these integrated methods and a lot of the lessons from his postdoc into this. And that is going to fundamentally change how people design cholesterol medications, just by looking at it and understanding. And so, I'm trying to impress upon you, that you can work in this area and then end up doing something sort of amazing in a totally different area. Okay, so setting the stage. So we have the infrastructure up. This is still early days. We're one of the few, groups that has the necessary infrastructure, Cryo-EM, the direct detectors, the know-how. And so it was like, okay, we've done HIV envelope, what else can we do? And so again, turned to Richard Feynman, and he said in 1990, prepare for the worst, right? $10 trillion was spent on the Cold War, and we never got invaded by Russia. Why don't we apply the same logic to climate change or to pandemic preparedness? There's a more recent TED Talk from Bill Gates, basically saying the same thing. If anything kills 10 million people in the next few decades, it's not missiles, it's microbes. So why are we not doing more in this area? Because as Jamie said, something is always going to come back. Infectious disease is going to be a problem that we always live with, and we want to get ahead of it. So that was somewhat of the inspiration. And then the other inspiration was sort of, this serendipity. So Jason was a young faculty at Dartmouth. He was working on RSV vaccines. I was working on HIV vaccines. We said, hey, let's do something together, in the backdrop of MERS and SARS outbreaks. And then I was fortunate enough to have Rob Kirkdorfer, who was a postdoc, who transferred to my lab and said, I want to work on coronaviruses. Let's do this. And I said, well, we don't do anything in coronaviruses, but we can give it a try. And so I really like this because I made a deal with Rob. Rob wanted to work on the inner parts of the virus. I said, you're going to work on the vaccine and the inner parts. And I said, this is like amazing that I wrote this email. Like, we're huge successful. We publish papers, whatever, and then we drink beers. Rob gets a faculty position. It's all good. And that was, it's sort of a joke, right? Because again, early days of Cryo-EM, people weren't taking it very seriously, but Jason recognized the potential, and really few people were working in this space and understood how to do it. So we ran it forward again, right? He sent us some MERS or SARS spikes. They didn't look good. And then we said, well, let's look at some seasonal coronavirus spikes that are related. So HKU1 came along, and it was like so obvious. What do? Take my daughter out and then put the sample in the microscope. And so this was in 2015, 16, and this was where we were in terms of speed. We really pushed hard on this. And so from May 7th, when the first samples arrived, to the first Cryo-EM maps, you can see in the bottom, it was only a couple months. And from there, it's again that Rob and Chris and others interpreting the structure, building the model, submitting a paper, submitting a grant, doing all the things. And this was like fast for back then. Now we solve structures in hours. But again, this was set the stage and really provided blueprints, right? So that's what we have. We have these engineering blueprints on this spike protein. And we say, okay, this is how a seasonal coronavirus spike looks like. How can we extrapolate those details to pandemic, like MERS or SARS at the time, to stabilize them in this pre-fusion conformation? So if you were to just do this randomly, there's about 25,000 possibilities of mutations you could make, which are totally not a reasonable amount of things to do. So the structure allowed us to focus in. We knew where the weak spots were, where the springs were that wanted to pop open, and we, after about testing 40 or 50 mutations, converged on these 2P mutations. that really allowed us to then produce these MERS and SARS spikes to study. And I think, really importantly, for both structure and for vaccines, you can notice when we tried to make MERS and SARS before, there are these little blips. If you get little blips, there's not much you can do with that. But as soon as we added these 2P mutations, we got a huge increase in the amount of protein. And so when you inject RNA into a person, they make a heck of a lot more antigen. That's a good thing. And it's antigen that's been stabilized and designed to be in the right conformation for antibodies to target. So again, working over here in seasonal coronavirus land led us to this. And then we published and patented the 2P. And so they were on the shelf when the pandemic hit. And so all of the companies that developed vaccines included these 2P mutations in their vaccines. And it always was beneficial to have them over not having them. So nature would have never put these there. You needed to rationally engineer and insert these mutations to get the effect. And vaccines can be delivered in lots of different ways, and all of you have had versions of different vaccines in this regard. As I mentioned, you can inactivate a virus and inject it. That doesn't work so well for envelope viruses. You can make it as a protein, and Novavax did that, and Novavax has a vaccine. You can vector it in a different type of delivery vehicle, a different type of virus, or as famously was done, encode it with RNA. But all of these things, the main goal of independent of the delivery was to deliver the stabilized spike protein for the immune system to see and generate antibodies. Another shout out here to Haley Perrett. She is an amazing scientist and illustrator, and she developed Science Simplified series during the COVID pandemic. And I encourage everyone to reach out to our website and check out these videos, because they really kind of distill a lot of the aspects. That's Haley popping champagne at her thesis defense. And I also want to acknowledge Haley and I went back and forth, and she helped simplify and beautify a lot of my slides. So I really appreciate that she's still on retainer and helping, even though she's in the startup biotech world. Okay, so I think the main message, right, is that all the tools were available, and that's why a COVID vaccine could be made so quickly. Right? If we start all the way over, modified RNA has been worked on since the 70s. It was the subject of a recent Nobel Prize, Katalin Karikó and Drew Weissman. In fact, the last time I was on this stage, I was introducing Katalin when she was here for a lecture. And they figured out that you could make RNA with these modifications that allowed it to evade immune recognition and not get degraded. So it became a viable delivery vehicle, and then Acuitas and others developed these nanoparticle or lipid nanoparticle technologies that made it so you could deliver them. We did, of course, these stabilized and engineering 2P mutations. And then the other sort of key thing that often gets glossed over is sequences, right? So I mentioned databases earlier. Early in the pandemic, Gisaid and Nextstrain, others were in real-time sequencing viruses and the spike protein. So such that if you were at a company like Moderna or BioNTech, you could download the sequence, you could add the 2P mutations, you could formulate it with what was known, and that's why RNA went really, really fast. So everything was poised in the right direction to do this successfully. And I think that's just a huge testament and win for the idea of basic research, right? A lot of the work that was done on modified RNA had nothing to do with vaccines, right? It was to deliver proteins as therapeutics. A lot of the work we did was kind of like, gee whiz, we like to engineer proteins and coronaviruses, nobody's working on them right now, so it'll be fun. And then a lot of effort again in these robust databases. And then the sort of next level result is it really opened up RNA as a platform. RNA was being developed for cancer and some other applications, but really the COVID vaccines now have opened up the floodgates for what can be done. And an article from a couple days ago from Wired basically is really nice and showing how getting through and getting an effective vaccine for COVID is now opening up the door for lots of different things like cancer. And has provided a lot of safety data and other data that'll shortcut a lot of the programs that people have been having on hold. Another little cool vignette just to acknowledge the team here. So Rob, Hannah, Chris, and Jesper were part of this. We were part of, again, a village of people that were acknowledged in a Smithsonian Institute display, I don't know, probably a couple years ago, but that was really nice to go see. Again, in retrospect, many of the people, on these slides weren't directly working on a COVID vaccine, but were doing things that all came together at the right time. Okay, so Rob's research arc, again, I said Rob came to my lab to work on these. These are the inside proteins, these polymerases. He, in his spare time, worked on the COVID spike and the vaccine aspects. And again, just to impress upon you, right, so this is just a citation metric, right? So this is how often the work is being cited or acknowledged by people in the field. And you can see, even though we published the first spike structure in 2016, it kind of got not a lot of attention. But immediately when COVID hit, it got a lot of attention. And same thing for these proteins, these polymerases. When Rob solved this, these are now the target of small molecule antivirals. And Rob's built a program around that. And again, you can see it didn't get much attention until it got a lot of attention. And it really reinforces the basic research aspect of what we do and how it can come to fruition. Okay, so now where are we? How do we make better vaccines? We do it rationally, right? We have better tools. We can image, we can understand, and we can do it faster, right? So we can take the immune sera from an infected person or a vaccinated person. We take out all their antibodies. We do a lot of analysis. We sequence the tens of millions of antibodies that are there. We do a lot of structural analysis. We do a lot of redesign, and then we iterate. And the faster you can do this, the better you're going to do in terms of a vaccine. This was sort of born out of necessity. And again, a lot of our HIV work. And so this is our polyclonal epitope mapping. And simply put, it's instead of solving one structure at a time, take one antibody, one antigen, it was take the antigen, mix it with all the antibodies, put it in a microscope, and then each one of these is a complex of a spike protein or an envelope protein plus one or more antibodies, and then use computational methods to sift through all the heterogeneity and mess. So Hannah, who's in the lab, Lars and Matteo, a postdoc and faculty here at Scripps, help bring this to fruition. And so now, instead of being a structural biologist waiting months for people to give us antibodies and things to look at, we were looking at them basically in real time. So we're running through that loop a lot faster and diagnosing, are we going in the right direction, in the wrong direction? And to give you sort of a comparison of how you would have done that before 2018, right? Each one of these is an animal, and each one of these is a mutation in a virus. So somebody had to make a mutation in a virus and then test whether it had a positive or negative effect on the antibody binding. And so the supplemental data of a paper would just go on and on and on and on and on. And this is useful data, but it's months of data and a lot of ambiguity and thresholding, whereas in a matter of a couple days, we could generate this. And this tells you all the things you wanted to know about where structurally those antibodies were binding. And that is really accelerating what we do and quite complementary to all of the things that we've done. Another fun research arc, Sandhya Bangaru, who came to the lab in 2018, she said, I don't want to work on flu. There's too many people working on flu. I need something new. And she said, how about coronaviruses? And I was like, perfect. Nobody's working on them. We just solved the structure. It's going to be fun. Of course, you know what happened. And so in her time, she then pivoted and she solved, actually, this is the structure of the Novavax vaccine, confirming that it looked the way it should. She also did a lot of this, what we call molecular serology or surveillance of pre-existing immunity to seasonal coronaviruses, understanding the set point at which people that were exposed to seasonal might then respond to coronaviruses, and a lot of beautiful work. And now, in addition to sort of running the COVID team in the lab, she's also building up her own program and understanding the molecular basis of peanut allergy, right? So something completely different that we haven't worked on, but by using these same tools that we use for HIV vaccine, shedding light on allergy and really filling it in. When she started, there were only a handful of structures available and really not a lot of information. So you can't do design, you can't do the things you want. And in the matter of a year, she's really transformed that and built up this really cool program using these tools. So as I said, we've created a lot of tools. This sort of assay that we developed is now used as an exploratory endpoint in a clinical trial, which is a big deal. And Gabe in the lab, who's been with me for most of his career, has really professionalized this. And we're part of a lot of different clinical trial studies and clinical networks to analyze immune responses to HIV vaccines and flu vaccines and COVID vaccines. And it really is the key information. Like I said, we're doing incremental and molecular medicine here, and we're trying to understand at atomic precision level, do the vaccines we design to be these wonderful, beautiful things elicit the types of antibodies we want? And in order to do that, we need the sequences of antibodies and we need the structure of these antibodies. And so it's become really fertile ground now as an assay on the back end to iterate more quickly. And then just a couple slides in closing, right? Vaccines are awesome, right? In ideal cases, the measles, they elicit durable, long-lived immunity. You never have to think about it again, and you can even talk about eradication. But what if you're immunocompromised or elderly? Then the antibodies themselves are good. We need antibody therapies, and a lot of antibodies, 20% of new drugs, are being developed for cancer and other space as well. So we said, can we invent a way to make antibodies faster and more informed? So just the details don't matter, but here's one of the a Regeneron pipeline for making antibodies. You're making hundreds of antibodies. You then have to characterize them, and eventually you come and say, will you solve the structure? We say sure, because the structure is useful for these rational design and improvement pipelines. But we said, let's shortcut this and go right from a drop of blood into the microscope and see what we can see. And so now, here's 20 different antibody structures from a drop of blood from Shiu, who's now at Moderna, and Gabe. And again, this allows you to get to structure faster, which allows you to do design faster. And then sort of the ultimate culmination of this, again, a little research arc. Alex, who grew up in war-torn Serbia, lost his dad and his brother, came and was just a remarkable person and scientist. He now is a professor at EPFL. And I basically said, I want you to get here. I know you can do this. I know you can take the data we generate in the microscope and then infer the sequence, which is a very difficult thing to do. And it took a lot of math and bioinformatics and other things, but he figured out how to do that. And so now we're making antibodies directly through structural methods and shortcutting a lot of the intermediate steps and down selection steps along the way. And so this is just highlighted, and really, here's again, it's not just one person. Rob, Chris, again, Charlie, Gabe, all contributed different aspects of this and make this work. And this is really ramped up, and we get lots of requests from academic collaborators and industry to apply these technologies to what they're doing. Okay, so just in the last two slides, I just wanted to complete the arc here, right? I think, again, all I've done is because of where I am, Scripps, the people around me, the mentoring, the grad program, and really the senior colleagues shaping me from an uncouth whatever grad student to a semi-couth person presenting you today. And we've been fortunate to receive royalties for the 2P work on the COVID vaccines, and now it's really about giving back. So, in addition to the science we are, as Jamie said, a training institute and a vector to recruit and then launch the scientists that are going to solve the problems of the future. And so with some of the royalties, I established a fund really inspired by Alessandra. So also pictures Julianne, who runs my flu team and is helping with a startup company, an amazing story in her own right. Alessandra started as a high-tech high intern, did a 5-week internship, and then contacted me after she graduated high school and said, hey, can I have a job as a lab helper? And I said, sure. And then she has just turned into this amazing scientist that goes to conferences, presents, is published. But she's working 2 jobs. She was going through community college and just getting by. And I said, this is crazy. We want to accelerate her. So starting this fund is going to help pay for her going to a proper 4-year college and open up doors that otherwise wouldn't have been there. So it's really that we bring these people in, we expose them, and then we really give them agency and support to succeed. And then with some of the royalties, and matching funds with the support of the Skaggs family and Pete, we were able to start some named endowed fellowships for graduate students. And this was really sort of personal to me because you notice there's now a Ron Milligan endowed fellowship. There's a Michael and Mary Reedy fellowship. There's some faculty that we've lost too early to cancer. Dan was on my committee. Ian, who was really fundamental to start the grad program and my own career, as well as his first student who died of cancer, and then some underrepresented minorities that contributed to aspects of antibody or vaccine research. And even though I have no say in the matter, I ended up getting Jordan in my lab as the Ron Milligan awardee, and Jordan's doing amazing things and basically building a program or bringing a program to my lab on malaria vaccine research. And it's really exciting to see where he's going to go and where all these recipients of these awards are going to go, because it really is just part of this cycle. And we really are in such a good place right now, and we should be doubling down on everything we're doing, including training, because the world in the future is a better place with these people out there conducting science and getting your support and help as well. So with that, I will end and say thank you.
All right, thanks a lot, Andrew. So that was a good drink from the fire hose of vaccine discovery. So we'll think about your questions, but let me just start out with one. So I do structural biology. I've done it my whole career, and you pick a problem, you got a molecule, and it misbehaves. So it's time honored that you make mutations to try to improve the properties of it so that you can study what you want. And so my question is, you talked about the 2P, which was key for stabilizing it. So you alluded to the fact that you made it and it made more protein, so it was more stable, and that was your goal at the time you did that, and then you got structures. So come clean, did you know that was going to make a better vaccine? Or was that something that you hoped because you have to have a stable antigen in order to get a good immune response?
Yeah, so I alluded, I think we knew from our HIV vaccine research that a more stable and higher expressing protein was more likely to give you a more robust immune response. So I think, again, that transfer of knowledge helped us believe that it would work. And of course, we tested it in animals.
So pre-pandemic, you tested those mutants to see if they were better antigens.
In the patent we filed, it has mouse immunogenicity data.
Immunogenicity, so yes. Yeah, right.
So you got to test it.
Yep.
I mean, designing is nice, but unless you drive the car, you don't know if it works.
Yep, good. Okay, so do we have any questions out here? I'm sure there are. There's one up there.
Not necessarily a scientific question, but I got the feeling that there was a lot of skepticism in the public as far as the vaccinations. Do you think it was paranoia, politics, ignorance of science, or you didn't deal with it?
Yeah, I mean, I would say it really is misinterpretation or amplification of sound bites that have no basis. I think, we need to do a better job just educating people, our children, to just have a basic level of science education. And, as scientists, we really seek out ground truth. We work with clinicians who care a lot about health and safety. And really their voices were muted and the skeptics were sort of amplified. And then it became just a social media game, honestly. The safety is amazing. The safety profile for these are ridiculous, in fact. Yeah, I mean, that's a problem, right? I mentioned herd immunity. The more people that get vaccinated, the less likely the virus is to spread. And then really, the unfortunate part is there are people that took the vaccine that were immunocompromised or elderly or had some other immunological dysregulation, and they became susceptible, right? It's like secondhand smoke. If you're smoking, you could be impacting the people around you. If you're not vaccinated, you could be a vector. You might not get really sick, but you could be a vector that kills a loved one or a random person on an airplane. So I think that demands you to go into it. I think what was not appreciated is that RNA has been worked on and tested clinically and has, again, a huge safety profile that had years of clinical data behind it. So there was really a concern that it would have a downside as a vaccine.
Another question? Yes, we have one down here in the front.
Can you repeat the questions?
Yeah, sure. We'll repeat so you can hear.
Thank you very much, Professor Ward. So I'd like to know how Cryo-EM/Cryo-ET overcomes the issue of specimen flexibility. I know you mentioned that in crystallography, you do need to crystallize the sample, and that can be difficult for flexible specimens, for example, glycosylated specimens, for instance. But I believe in Cryo-EM or Cryo-ET, you also do need to vitrify the sample. So how exactly is the flexibility addressed? Is it related to post-processing, computational processing? Is it averaging? Just like to know more about that.
So in crystallography, you get one structure from the crystal. Every copy of the protein or complex is the same, and you get a single structure. For Cryo-EM, you get all of it, right? So again, if we go back to the hawk analogy, you get all the pictures of the hawk in flight, and then you use computational methods to basically sort them into bins and say, here's wings up, here's wings down, here's wings halfway, and then you can average all the ones that are the same and get high resolution. And so a lot of the value in Cryo-EM is not just the single static structure, but it's the multiplicity of structures and the wiggliness of biology that you're able to observe.
So Andrew, you showed the HIV vaccine that you participated in, and so that arc was 15 years. So take me through it. So the phone rings tomorrow, and it's the CDC, and they're like, we have this virus, it's an outbreak, we need antibodies. Take me through the arc of how long it would take you to get there.
I think it's pretty fast, and I think, I didn't even mention it, but a lot of the new tools for AI and AlphaFold and predicting protein structures allow you to do design really quickly. And so I think all the elements are there, right? I think RNA remains the fastest way to get a vaccine in people. That's months long. As long as you know the structure, and again, if it could be stabilized, that's useful. And then you need the sequence. So it really, we've reached a place. Now, the real problem, even if we can go fast, right, is actually reaching a lot of demographics, being prepared to manufacture at scale and across the world, right? Because what happens in South Africa actually is going to impact us, right? So if we're only immunizing us, then South Africa is an incubator, and our vaccine might not be as good because the virus has evolved and changed. So we just lack the infrastructure. And I think the other important thing is, like colleagues like Christian Anderson are doing surveillance, right? Looking out for the next bug, raising a flag to say, hey, we should look at this, we should be working in this area because it could come up. And that's what's happening, with H5 and cattle flu and other things. Surveillance is a really important aspect that's really underappreciated in terms of the epidemiology and knowing when to start on a project.
So one of the things I was wondering, so now you can basically take blood from a person and then collect all the antibodies that bind to some antigen. So is there any merit to using that as a tool to assess someone's immune status? Have you been exposed? Do you have the right antibodies, or are you susceptible?
That I've put that in a grant, and I've advertised that. That's what I want to do, right? I want to have, instead of a COVID test where you see a little band, I want to have, COVID, flu, RSV, all respiratory viruses, and we just take a drop of your blood, we image it and say, you've probably had this before or been vaccinated against this. Here's the antibodies, and here's at a population level, are you in a good place? Are you in a bad place? And yeah, that's how I see this technology progressing.
Yeah, and following that, seasonal flu is a challenge because the spike proteins on the flu change every year. But it seems like we don't really factor into it in making the vaccine for this year what people actually have. So if you not only know what's out there, but know what people have in their systems, wouldn't that be a really,
yeah, agreed. More microscopes, more money, we'll do it.
Okay.
I think with flu's really interesting because some people that were exposed to, H1 60 years ago are protected against some modern strains, but younger people born in the 90s or 2000s aren't, because their original or their immune system was trained on something different. And so that's where you get into personalized medicine, understanding the demographics, your immune history, your exposure history can allow you to make bespoke vaccines for the different groups that would have maximum benefit.
Do we have, there's one up in the back.
I'm very interested in understanding more how you jump to using antibodies for immunocompromised patients and the elderly. So you skip the steps of the vaccine because of their immune status or the weakened status. And when you jump to the antibody to give to the patient, are there any rejection issues? Like, for example, if you have tissue transplant or heart transplant to a patient, I'm just curious about that.
Yeah, so, right, in a vaccine setting, or even if you've been infected, your B cells, ideally, for the rest of your life, are just pumping out antibodies that protect you. For immunocompromised people, their B cells don't work right? So they either don't respond at all, or they don't pump out antibodies for a long period of time. So you can give them antibodies as prophylaxis or therapy that are a surrogate. And because they're human antibodies, they're by definition biologically compatible. So there's no issues with tissue and transplant. There's issues in compatibility, but for antibodies, they're not. And that's why so many new drugs and cancer drugs and lots of drugs are being developed as antibodies, because we already know ahead of time that they're going to be safe and compatible with any human we put them in, essentially.
Okay, another question here.
You talked about personalized medicine and bespoke meds. How do you shorten the safety profile? How do you get the drugs in real time to people, given that you can figure out, okay, you need this particular protein to solve the problem or whatever? I mean, we keep hearing about genetic medicine solving all these problems, but isn't there a total bottleneck on the approval process, or how do you get around that?
Yeah, I mean, I think as I showed with that Wired article, a lot of the regulatory, red tape that slowed down a lot of those therapeutics delivered with RNA is significantly reduced because of the examples for the COVID vaccines. So just like an antibody, like I said, a human antibody that hasn't been modified that passes a few safety tests is likely to be safe and okay. RNA is sort of moving in that direction, that as long as we don't stray too far from the key elements that have been in people before, the safety window is likely to be very high.
Here.
Hi, could you tell us a little bit about what you've learned about drug resistance, both in infectious disease but also cancer?
Yeah, so that takes me back to my grad and postdoc days. Cancers or bacteria have these pumps that do normal function, but if you load the cells up with drugs, they start to use those pumps to pump out drugs, chemotherapeutics, for example, and then cancer starts mutating it so it can recognize and pump out those therapies even more. So cancer, just like a virus, right? So your immune system is using both the B and T cell arms to surveil all the time and remove cancer, right? Cancer is happening all the time, but you have a healthy immune system, it eliminates that. But we are subject to the cancer mutating and accumulating mutations enough so that it can break through, and then you're no longer resistant. That happens with aging and senescence, but it also happens because cancers mutate. So just like we should be always worried about monitoring infectious disease mutations, we should be worried about that. And that's where, again, a lot of personalized medicine comes into play, into predicting early, the surveillance early, and then tracking it, and then giving the right antibodies or therapies.
So maybe we should get out. It's after the hour, so let me just ask one last question. You described a couple of points where technology advances sort of provided the impetus to sort of really move forward. And I think a lot of what you described in the last part of your talk was, in part, there's AI that's involved that's enabling you to actually do this. So what's next? So where do you see this going? So give me your crystal ball for in 5 years, what are you going to tell me that's going to blow my socks off?
Good question. It might be that we're ready to sell some microscopes and use more computers, but
right.
But actually, no. I think in a lesson I learned as part of a startup we're participating in is you can design all you want, right? And AI and structure-based approaches get you to sort of the starting point much faster in days sometimes. But nothing replaces testing. Biology is really complicated. It's hard to model what's going to happen, and you actually have to have a robust in vitro and in vivo pipeline. And as I showed, a lot of our time and effort now, because design has become easier, is on analyzing responses, creating better tools to look in experimental settings. Are we on the right track? Are we on the wrong track? So I think really we're just going to start faster, but then we're going to need to really shore up our assays to make sure we're in the right direction. And then I think it really is, again, a village, right? Where the bench scientist understands all the processes and hoops you have to jump through. And so you can design with intent, and you don't have to remove liabilities or things you've accumulated because you work in this academic setting where you're completely unconstrained by real world reality of industrial manufacturing and clinical trials. So I think, the way I see it is a lot of the work we're doing here that's far afield is going to, hopefully in our lifetime be Alzheimer's vaccines and cancer vaccines and all these things that increase our health span if we get it right.
Okay, I think we want to. Can we put up the last slide to indicate the next front row? But I think Andrew, it's been a real romp. through structural biology, and it's been great to see you develop, and I hope you ignore all of my sage advice going forward. It's been a good strategy for you. No, it's just fantastic to hear about. So we're going to have a reception out here for everybody who's in the audience. Thanks for everybody who's virtual. Next front row, it's Luke Lairson on April 16th. And when you come to the auditorium, you will see me in my seat in the front row. Thank you.